Hi everyone, this is Dr. Jyoti Bala. I welcome you all on my YouTube channel. In this session, we will be exploring some of the wonderful bioinformatics tools, especially which are used in primer designing. So as many of our participants are from biotech and molecular biology background, along with the bio IT, they often require these primer designing tools. So let's get started. The number one tool is Primer 3. This one is used for designing the PCR primer. The key features of Prime 3 is it offers highly customizable setting for primer design, including the primer size, melting temperature, and GC content. Primer 3 also supports degenerate primers and multiplexing capabilities. The next one is OligoAnalyzer. This one is also utilized for analysis of oligonucleotide properties. This tool evaluates oligonucleotide sequence for melting temperature, secondary structure, and also for the potential dimer. It helps to ensure the design of primer that are suitable for PCR and other applications. The next tool is GeneFisher. It is an interactive PCR primer design. GeneFisher provides an easy-to-use interface for designing primer with a real-time feedback on primer quality. They support batch processing and include algorithm to avoid the primer-dimer formation. The next one is Primer Blast. This is utilized in primer design with specificity checking against the NCBI databases. The Primer Blast combines primer design with blast search to ensure the specificity to the target sequences, thus reducing the likelihood of the non-specific amplification. This integrates well with the NCBI vast sequence databases. Adding on the list, the next one is Primer Quiz. This helps in custom design of the primer and probe for PCR. This tool from integrated DNA technology allows for design of highly specific primers and probe tailored to the user-defined parameter. It is particularly useful for quantitative qPCR application. The next one is Fast PCR. This helps in designing the primer for various PCR amplification. This supports the design of standard, degenerate, and multiplex PCR primer. This includes modules for the in silico PCR, restriction analysis, and sequence manipulation, making it a versatile tool. Adding on the list, the next one is Patch Primer 3. This is a high-throughput primer designing tool. This is the extension of Primer 3, which is designed for high-throughput project, allowing user to design multiple primers simultaneously. It is especially useful for the large-scale genomic project and microarray studies. The next one is Amplifex. This one is used for visualization and management of PCR primer. Amplifex helps in user design, manage, and test PCR primer in silico. This provides visual tool to check primer binding sites and predict the size of the PCR product, aiding in the troubleshooting and optimization. The next one is Primer Premier. This is a comprehensive primer design and analysis tool. This one is commercial software which offers advanced algorithm for designing highly specific primer with minimum secondary structure and primer dimer formation. It supports a wide range of applications including SNP genotyping and real-time PCR. And lastly, the Primer Express. This one is used to design the primer and probe for real-time PCR. Primer Express from Thermo Fisher Scientific is optimized for designing primer and TACMAN probes, especially for real-time PCR. This provides comprehensive analysis tool to ensure the high efficiency and specificity of the assays. I hope you have liked the session. If you are having any questions or query or suggestion about these tools, or if you want to add some additional tool, you can drop the message in the comment section. And don't forget to like and subscribe the channel and do share these information among your scientific endeavor. Thank you.